God dag til alle, and I'm very honoured to be asked to speak at your meeting. Here is my title in Welsh, Capia Cwyr Campus Cymru, a little bit of alliteration. I should first explain a little bit about me and my country, and then introduce the wax caps. So I'm speaking from here, and you see, maybe you are familiar with this castle, and that's my house. I'm looking at the castle down the hill, but my workplace is here.
Yes, we see your mouse.
You see a mouse, yeah.
Yes.
So I live maybe 100 kilometers north of Aberystwyth in Caernarfon, which is where the castle is. And here is Aberystwyth, and here's our university. So both of these are small towns, and around them we have lots of countryside, but you don't see many trees. So we have a lot of grassland because my ancestors cut them all down. It's always difficult to know what other people have a perception of your country as a perception. I guess it's rugby here, our favorite sport, and we like singing and also digging holes. So most of the 100 years ago, there was a lot of digging of holes because of the interesting geology, because of the mountains. And I guess the mountains are one of the reasons why some of the grasslands that are on tops of the mountains or on the steep slopes didn't get ploughed. And that's why we have lots of wax caps here, just because they've been saved from agricultural improvement compared to areas where the land is more flat. I was looking back to see when people first started to notice wax caps, and there's this book from 207 years ago where this guy, who was a vicar, a preacher, he wrote this book about the Isle of Anglesey, which I guess was named by your ancestors in English. Anglesey Ynys Môn is what we call it in Welsh, but Anglesey, I think, is a Viking name. There's some interesting finds he made. This one, Agaricus dyleniae, is not known to many of you, but it was found. I'm interested in this because all my family are farmers, and I have one ancestor who farms this farm in Anglesey, and I've been to his fields, and him and his father, they ploughed all the fields. They're very proud of the fact they ploughed all the fields and made them productive, which is kind of a bit sad, really, because now this species, which we don't know what it is not there anymore. I think it's probably Entoloma bloxamii. So maybe this was the original name for that species, but it wasn't named properly or formally. So here you see our topology again, the geography. You see lots of grassland. We have a higher proportion of grassland than the other countries of the United Kingdom or the Great Islands of Great Britain. And we have a large number of sheep, many millions. But I guess after Brexit, nobody will want to buy them, so that number might go down. So maybe some of the farmers who voted for Brexit will have some regrets. Anyway, in the past 70 years, maybe 90% of lowland grasslands, probably across Europe, have been lost. But some of these steep slopes are difficult to plough and to improve. So that's why the natural grasslands or semi-natural grasslands are still there. And you can see they're mostly, the map of Great Britain, you can see mostly in the west, on the west side, where we have a similar Atlantic wet climate. And this term wax cap grasslands was defined by Yves Arnolds in Netherlands, where they have maybe only 200 hectare. So he said was left in the whole of Netherlands of this type of habitat. So it's a threatened habitat. And generally, whilst the Hygrocybe and the Hygrophораceae are the wax caps, the Claveriaceae, Entelomataceae, Geoglossaceae, and some of the Dermalomas are common there. And so we have this rather ugly name, Cheg D. It wasn't my name, it was my friend Maurice Rotherham who made this name, just to describe these groups that you find mostly in grasslands. But I see from looking at GBIF that Scandinavia, and in particular Norway, in the same westerly regions, has similar habitats and lots of finds of this fairly unusual wax cap near Hygrocybe ovina. So this is the person I have to thank for making me interested in wax caps maybe 20 years ago, Maurice Rotherham. He was a journalist. So he was an amateur mycologist, but he introduced me to them, and he also raised a lot of interest in the British Isles in these fungi. They were kind of neglected before. This is the man who started it, I guess Yves Arnolds, with his work in Netherlands. And I want to recognize the immense contribution of David Bortman here when he, 25 years ago, published the first genocide book. So this gave a stable taxonomy, so people could go out to the field and identify the fungi and all sing from the same hymn sheet when it comes to names. And more recently, we've done some of the molecular phylogenetics to make the names clear and work out which species are related to which. Something else we do in the UK, because when I started, people didn't know what wax caps were and didn't think that fungi were important for conservation, especially grassland fungi. So we did things here. Here's my friend Gary Easton with a hat. It's very dangerous to wear this hat, because if you turn suddenly, when you stop turning, the hat has a lot of inertia. So it's a dangerous hat. But you have these types of awareness raising, making people know about these fungi. And here you have in England, in Bristol, John Bailey, who was formerly a plant pathologist. He was an academic mycologist, but interested in microfungi. Now he's retired. He doesn't care about microfungi, only macrofungi. So him and lots of people like him make an important contribution. And I've also been trying to raise awareness of the fact that that fungi, it's not just animals and plants that need conservation, it's also fungi. One of the first big battles we had was in Cardiff, in the capital of Wales, and it's this reservoir here. And because the water for this city comes from higher up in the mountains now, they were planning to build houses, to empty the reservoir and build houses. But on the embankment, this green area here, in 6 hectares of embankment, they found 28 species of waxcap. So the Countryside Council for Wales, the conservation agency, gave legal protection. They made it a site of special scientific interest, SSSI. After a campaign here, this is the wife of the Prime Minister of Wales, who used to be, so it had important supporters. But the company that owned the reservoir, the lake, they were unhappy about this because they would make maybe 20, 30 million pounds from selling the houses. So they went to the High Court, the highest court in the UK, to challenge the decision. Unfortunately, they lost. Here you can see the decision of the judge here. So wax caps are more important than houses here. I guess if this decision had gone the other way, it's possibly I would be looking at other fungi and not wax caps by now. Our efforts aren't always appreciated. So here you see a headmaster a little bit annoyed that his new school couldn't be built on an old meadow. So one aspect of the work we've been doing is to try to establish what management conditions are the best for maintaining diverse populations of waxcap fungi. Sometimes people here think you have to cut the grass short all the time, but that's not compatible with the biodiversity of plants and insects, for instance. So when we did this experiment, we made a replicated design you can see here in a field which we knew had good waxcap populations, and we found that the best treatment, the most Cheg D or waxcap and allies fungi, most fruit bodies were found on when you let the grass to grow till July, and then you cut hay in the traditional manner. And then after that, you put sheep to keep the grass short in the late summer and the autumn. So this is good news, because it means that if you have a semi-natural grassland and you want to conserve all the biota, then hay cutting is a good way. But you can see also that if you keep the grass short, this is also good. And if you keep the grass longer, 8 centimeters here versus 3 centimeters, this is not so good. Possibly because the wax caps aren't stupid. They don't make fruit bodies in long grass, because the fruit body wouldn't serve its purpose. It wouldn't spread the spores. So I guess they save their energy for when the grass is shorter. I imagine they sense carbon dioxide levels as the primordium of the mushroom is forming, or something like that. So this is good news for conservation. But in terms of studying the ecology of waxcap fungi, they're not easy to work with. We can't grow them in pure culture in a petri dish in a laboratory. And even when we try to germinate the spores, this is as far as we got. And you notice these are the more common species of Hygropheraceae. So they make like a spore, the spore germinates and it doesn't grow very far beyond this point. And most of the species we examined didn't do anything. So it slows down the progress of research if you can't do some experiments in a laboratory. From the phylogenetics, from the family tree of the Hygropheraceae, we maybe get some clues. First of all, the fungi we call waxcaps are here. This is Hygrocybe. But some of them, like Cuphophyllus, are only distantly related within the family. And in between them, you have Hygrophorus, which is ectomycorrhizal. And also you have Lichen omphalia, so you have lichenized Hygropheraceae as well. So it seems that many members of the Hygropheraceae are biotrophic. So maybe this gives us a clue as to the ecology of the Hygropheraceae or the Hygrocybes we find in grasslands. Possibly that they are biotrophic. So one way we investigated this was using stable isotopes. So just in case people aren't familiar with this method, it's looking at the abundance of the heavy isotope of nitrogen, nitrogen 15, compared to nitrogen 14. These are not radioactive isotopes, they're stable isotopes. Similarly, with carbon 13, the heavy isotope of carbon, which is usually mostly carbon 12. And in a food chain in the sea, you see that these levels, the abundance increases of the nitrogen and decreases of the 13 carbon. So you get this progressive change along the food chain. So when we apply that to, so it just shows the trophic level. So maybe this will give us a clue from looking at the wax caps. So here we find the saprotrophic fungi. The red ones are in woodland, the species growing on soil in woodland. And here are the ones growing on soil in grassland. What you find is that the wax caps are here. They have very elevated levels of 15N. The delta 15N level is high. And the carbon, the delta 13 carbon is very negative, is low. If you look at the ectomycorrhizal fungi here, in blue, you see that they're also distinct from the saprotrophs, and it's almost like the wax caps are extreme in their isotopic patterns. And in Europe, we find the wax caps mostly in the grasslands. And I saw from one of Jon Bjarne Jordal's papers that you also find them in woodlands in Norway, and we find them sometimes here. Outside Europe, mostly you find the wax caps in woodland habitats, but even in the woodland, they have the same unusual patterns of isotopes. So this gives us a consistent clue globally for what they, that they have some unusual nutrition. So I think is that they're mycorrhizal, but I acknowledge that I haven't found the convincing evidence yet that this is the case. We see them in the roots. You can see here that Hygrocybe, there's a small hypha with a clamp connection. Here you go, inside the root hair. And by PCR, we can detect the presence of these fungi in plant roots. But if you try to follow from the base of the fruit body into the soil, you have no success. With ectomycorrhizal fungi, you can follow sometimes the hyphae back to the root. I tried lots. Maybe I'm just not good enough at doing it. But if it's not possible, it's maybe possible, but not for me. So we have all these little clues. The unusual profile of the isotopes. We know that if you add fertilizer to a soil, that kills the wax cap. So maybe it disturbs the balance of any mutualism with the plant hosts. We have one finding here that's interesting but not yet explained, is that if you kill the soil animals, this inhibits the fruiting of the wax caps. So it's possible that maybe they're deriving nitrogen and feeding it to plants from the bodies of soil animals, but we didn't show that yet. In terms of the other way around, the other part of the mutualistic interaction, if they're mycorrhizal, if you feed 14 radioactive carbon dioxide to a soil, you find that to a plant, to an area of grassland, we found some accumulation in the wax cap fruit bodies, but the amount was the sample number was quite low. What's unusual with the wax caps is that apart from Agrostis and Festuca, which are almost in every grassland in Northern Europe, we don't think there is any specificity. But if we add weed killer, herbicide in patches, we don't have fruiting. So I have lots of small pieces of my jigsaw, but the jigsaw isn't complete yet. What we need to find is the mycorrhizal interface to find what exactly is happening here. So the other technique of modern science that we have used a lot now is using eDNA metabarcoding. Just like Håvard was telling you earlier with the dust samples, this is a good way to find not just the fungi that grow, but also the ones that don't grow on a petri dish. And I'll just briefly explain what we do. We pick a piece of grassland, 30 by 30 meters squared, and we collect from each one 36 soil cores like this. We treat this as one sample. So this one sample is the average for this area of grassland. We freeze dry and we grind this soil very finely, and then take a small subsample, maybe only 200 milligrams, from the dried ground soil, extract DNA, do the PCR just like you would do for the normal DNA barcoding, but on the whole population. And then we do the DNA sequencing. There's lots of different devices. We have nearby us, we have this machine called an ion torrent. It's designed for human genome analysis. So it's very fortunate that fungal ecologists can also do interesting stuff with these machines. We use various databases, RDP for the large subunit part of the RNA, ribosomal RNA operon, but also the more familiar ITS2. And we use the UNITE database for that. So we're very grateful to Urmas Kolyalg and all his colleagues in Estonia for making this database. And I think there was lots of contribution to its development from Norway and Sweden as well. So this tells us, it gives us a species list and also a reasonable idea of the relative proportions of the different fungi. So when we go back to the reservoir clinician and compare the fruit body data with the DNA data, we have good correlation. Here you can see some quadrats that were distinct here, also being picked out here in the fruit body and the eDNA data. What I find interesting is the fact that of all the fungi in the soil, in these habitats, the majority, the dominant species are the Cheg-D fungi. And it varies. In this case, the 70% of the DNA in this quadrat, so 70% of the biomass is from Hygropheraceae species. We get a slightly different ratio with the ITS2, slightly lower. But we consistently find this in sites where there's lots of wax-capped fruit bodies seen, that they are the dominant fungi in the soil. Usually people talk a lot about the arbuscular mycorrhizal fungi being very important in grassland soils. We think they are important. I think maybe they work harder. So there's less biomass, but it works more hard. The wax caps are maybe a little bit lazy. So it's nearly impossible to get funding for this type of work from the research councils in the UK. But I found it was possible to get work from conservation organizations. And I'll give you, this is my favorite example, is this land is in the middle of Birmingham in England. So it's surprising that you have any wildlife site here, because the whole global industrial revolution began only 3 or 4 kilometers away with the steam, when they invented the steam engine. James Watt, I guess you heard of him. So you have this little island of biodiversity that in the middle of this huge city. And these people, this guy was a student in Aberystwyth, so he knew about the work I did. He asked me to come and have a look to take soil cores to look at the eDNA here. And you can see that in green are the quadrats we took. And this guy, Nick Williams, had been studying the wax caps here, and he found 22 species in his 20 years of studying. So it was obviously a good site. That's a good level of biodiversity. So we had a look at the eDNA from these different quadrats. So this is like a species list. Here's the species. And you can see quite a lot of them here where it says FB not found. That means fruit body not found. So we found what we thought were novel species. These numbers are the number of sequence reads from the high throughput sequencing to give an approximate idea of abundance. I didn't put the Entoloma here. I was talking with Tor at lunchtime about how the taxonomy of Entoloma is really hard, and I'm really pleased to see that Machil, Nordalus, and I think several of you in Norway are making contribution to updating the molecular phylogeny of Entoloma. So the aim with this site was to give it legal protection, to make it a site of special scientific interest. And in our eDNA work, we identified 6 species in the soil DNA that were not yet seen in fruit bodies. So I could tell people that if they looked in this piece of land, they could find Hygocybe cutaconica, and here Hygocybe intermedia, and here Hygocybe spedicea. So when they went to look later, the autumn after, they found these species. So that was really encouraging. This is eDNA guided fungal surveying. And also this one, Microglossum olivaceum, we found here. This was important because this one has special legal protection in the UK. It's a super rare fungus. So we found of the 6 missing species, they were able to go out afterwards, and when you know what you're looking for and you know where to look, it's easier to find things. So they found 4 of the 6. So we increased the count. We increased the species count to make it more certain that legal protection would be granted. So I think this is the first time ever that anybody's used eDNA evidence to help achieve legal protection for a site. And you can see that here's the site, here's the area we looked at, and this whole area is now the protected site. It includes a golf course here, so we also looked here and we found 2 additional species. The reason why we had to go and get the eDNA evidence and then afterwards go out and look for the mushrooms was because the eDNA evidence isn't admissible evidence for legal protection at the moment. But maybe in the future that will hopefully change. The other way we've managed to get some funding for doing the research is to help with planning applications. So here, this is just south of me now by the seaside. You can see this, the Cardigan Bay, this area of sea. Aberystwyth is kind of there in the distance. And on the coast of Wales, we have lots of these audible caravan sites for holidaymakers. And this field is just the other side of this woodland. And this is a small golf course that wasn't being used very much. So the company who owned the caravan site wanted to put caravans here. But before they could do that, they had to apply for planning permission. And part of applying for planning permission is to do a biodiversity survey. So this was the first time in 2013 we did this, when we went and I sampled the soils like I described. And we found some wax caps, but we compared them. Here is the golf course. Here. You can see here I took samples from the areas where there's some birch trees and heather, and they clearly have different fungi in the soil. And here are some sites. Here are some sites with known high levels of wax cap populations. These ones had wax caps but weren't distinct. They weren't special. So unfortunately, the planning permission was granted. We've had other cases where we've managed to find rare fungi and they refused planning permission. So this is good news. This helps to protect these precious wildlife sites. The other thing we do is to help the Welsh government with, there's a law to do with changing the use of land. It's called the Environmental Impact Assessment, EIA regulations. And here I have an example of a farmer down in South Wales. This is the picture before. I didn't yet get a picture afterwards, but essentially he ploughed this area to plant wheat, like he did on all the fields surrounding. And he denied it. It's kind of obvious if you look at an aerial map that it used to be a heathland here. But we could, by obtaining soil samples just a few months after he ploughed it, we could see that we could find Lepista enteriza, which is a fungus associated with juncus, and some wax caps in this area. So that helped the government prosecute the farmer for illegal change of land use. So I can't do the stuff I do. I'm stuck in my office most of the time, just like Hovad, I imagine. I'm very reliant on my friends who are out in the field all the time, and many of these people in the UK are retired people, and the citizen scientist is one word. The most effective one is my friend David Harris, and he lives in Pembrokeshire in southwest Wales, and he organises the Pembrokeshire Fungus Recording Network, and they have made these various guides, which are very useful to help get new people interested in the work. And he also then, I taught him how to do DNA extraction and PCR using this device. It's called a Bento Lab. I think it's a company in London. So this is them. They collect the samples. They can extract DNA. This device lets them make a gel, and there's a UV box here. They can make an agarose gel to see if their PCR worked, and then they can send it. They send it to my colleague in Aberystwyth, Karen Evans. She runs the sequencing lab. So she I think maybe £3, 4 euros per sample. And she sends them a sequence. So they do this separate for me now. But they kind of quite like it. They have a lot of fun doing this kind of biohacking, I think is the word. And also it was assisted by one of my former PhD students, Brian Douglas. He got a job in Kew, in Royal Botanic Gardens Kew in London, and he promoted the use of the Bento Lab through the Lost and Found Fungi project. And now I thought I'd ask some questions of you, the audience. First of all, I'm not quite sure what term is the best one to use. In the UK, much of the scientific or the taxonomic expertise on fungi now is with people who aren't paid to be mycologists. It's kind of quite a bad situation. So we're very reliant on the field mycologists, as we call them. Sometimes people call them citizen scientists, and some of them don't like that term, or amateur mycologists or paraprofessionals. So one of my questions was, for many of you in the audience who are, you study fungi for the love of fungi, what do you like to be called? Another question would be that in 2 years' time, IMC12, the International Mycological Congress, is being held in Amsterdam. I understand that Hovard is one of the keynote speakers there, so that's another reason to come. But the other reason, a second reason to come would be maybe to meet other field mycologists from around Europe. It's not often that this conference is held in such a convenient location for us. And my last question was, I'm working with David Harris. We want to try and produce a key for the earth tongue fungi, the Microglossum, and then also the Geoglossaceae. So if anybody has samples of especially Microglossum, which is a very rare species, not often found, I'd be very grateful if anybody had any. I'm happy to DNA barcode them for you. And if you have samples yourselves, my friend Karen Evans, who runs the genome sequencing facility in Aberystwyth, would be happy to receive samples from you. I think I need to double check with her how to do that, but I understand that quite a few people in the UK now use her. And my last question was here. I only just found this last week. Here is the GBIF map for near Hygrocybe nitrata, and I've put the picture here in an inconvenient place. It's very unusual in the UK and Wales, but I see it's common in Norway and Sweden. And when we did some work with the eDNA with Slavo Adamczyk in the mountains, in the Carpathian Mountains in Slovakia, we found this was very common there. And the only reason I can think of is that both places you have a very hard winter where the soil freezes really hard in the winter. I find this very interesting because this is giving a clue as to why certain wax caps occur in some places and not others, because that's been difficult so far to be able to make progress on that, and finding what niche differentiation there is between the different wax cap species. And to finish, I'd like to thank the people who funded us and all the people I worked with to do this in the lab. These are the funders in my lab, especially Andrew Detheridge, and then all the citizen scientists or field mycologists, and also the people who look after the field sites where our experiments take place. So thank you very much for your attention.